Basic principles of regulation of gene expression in prokaryotes. So let us ask a question. If there are 25,000 genes, say for example, in a prokaryotic cell, do we basically observe that at a particular time of that cell, do all the 25,000 genes express themselves at the same time? Or is it that some genes are expressed at a particular time or some genes are expressed at a different time? So the answer to this is that certain genes that are present in the prokaryotic cells will express themselves only on requirement. That means only when the cells require a particular enzyme or a particular protein, the genes of these enzymes or proteins get activated or they get inhibited. So there are certain mechanisms by way of which these genes are regulated, and this session is going to look primarily at the principles that are involved in these mechanisms. So what are the learning outcomes of this session? Well, there are defined mechanisms that can increase or decrease the expression of genes as per requirement of the cell at different stages. In prokaryotes, most of the regulation is observed to be at the transcription initiation. When the RNA pol is just about to begin transcribing a gene, that is when you can have the transcription stop, or that is when you can have the transcription activated. So this mainly is at the isomerization step of the transcription, which is the commitment stage, where once the RNA polymerase moves from the closed complex to the open complex, it gets committed to transcription. There are mechanisms of regulating expression post initiation of transcription as well, which means that the transcription has already begun, but immediately after the transcription has begun, it can be regulated to ensure that that the expression is to be continued or to ensure that it is stopped. So it can take place post initiation as well, and to a great extent, especially in the eukaryotic systems, it has been observed that control extends right till the post-translation mechanism included. There are mainly two types of regulatory proteins, which are positive regulators or activators, or negative regulators or repressors. So we will look at now the principles or try to understand the principles of transcriptional regulation. So let's consider we have a bacterial genome, and in the bacterial genome, there are certain genes that are called as housekeeping genes. What does this mean? It means that these genes are expressed within the cell at all times. So these genes are mainly having promoters that are called as constitutive promoters, in the sense that these promoters can be bound to by the RNA polymerase and the initiation can start anytime, and these promoters are also more accessible for transcription. Now, there are certain other genes which are regulated genes. It means that these promoters are the regulatory promoters, and these promoters can be bound to by activators or repressors, that means the regulatory proteins, to basically influence the expression of the gene. So we are looking at the fact that if you have a DNA, from DNA you will get RNA. So that is what is called as transcription. And you know that the RNA pol always binds to the promoter region, starts the transcription, completes the transcription to basically give the RNA. Now this RNA is then translated into its respective protein. So in this central dogma, where are the control points of regulation? So the control points of regulation are mainly at the start of the transcription, and then it is basically after the transcription is done during RNA processing, or also at the translation, before the translation begins. And of course, one can also see the control points post translation, where the protein can be degraded. If the cell does not require the proteins can be degraded. So that is also a point of regulation. But in prokaryotes, majorly, the focus of regulation of gene expression is at the start point. That means starting of the transcription per se. So the regulatory proteins, as just mentioned, there are two types. One is the activators and the second is the repressors. Both the activators and repressors are proteins that specifically have what is called as DNA binding motifs. So for example, there can be helix turn helix, or they can have zinc fingers, or they can have talons. So these are certain motifs that are present in these proteins that can interact with specific sequences in the DNA. And those specific sequences in the DNA are the cis elements, and these activators and repressors are the trans elements. Now, let's say, for example, you have a DNA where you have the promoters. So, one is the -10 and the other one is, say, for example, the -35. So, we all know that the RNA polymerase comes and specifically binds to these promoter regions through its sigma subunit. So, the sigma 4 binds to the -35 and the sigma 2 binds to strategically the -10, which is a Tata box. And the Tata box is an AT rich region. So we know that if it has to open, an AT rich region is more easy to unwind than a GC rich region. So strategically, the -10 is an AT rich region. So this closed, when the RNA polymerase binds to the DNA at the promoter region and it binds stably but does not unwind the DNA, then that complex or that conformation of the RNA polymerase is what with the DNA is what is called as the closed complex. But when you have the RNA polymerase changing its conformation in such a way that also the DNA opens up at the -10 to form the transcription bubble, then that is what is called as the open complex. So this step of the RNA polymerase taking transcription from the closed complex to the open complex, this step is what is called as isomerization step. Why are we stressing or focusing on this step? Because it is well understood that once the RNA polymerase forms an open complex, then it is committed to transcription. It is going to escape the promoter and begin synthesizing the RNA using the DNA as the template. So this is important. So if at all one has to look at controlling regulation, then it is at this step. Because if it goes from closed to open, after it is open, it can only move forward, it cannot go back. So the way to actually control is to ensure that the closed complex does not go to the open complex if you want to prevent the transcription. But if you want to initiate transcription, then you have to ensure that it goes from the closed complex to the open complex. Say, for example, an activator binds to its cis element and it interacts with the RNA polymerase. Then what is observed is generally the closed complex goes to the open complex, and then you will have many RNA being formed. So therefore, this is a positive regulator because it is giving rise to many RNA from the gene. But on the hindsight, if a inhibitor comes and binds or a repressor comes and binds, and it binds in such a way that either it will not allow the complex to go from the closed to the open, or what can happen is if it allows, then it will only allow synthesis of very few, because it is going to make the conformation of the RNA pol such that its affinity to bind to the promoter is not strong, and hence it may not go forward with transcription as many times. So the amount of mRNA formed is going to be less. Now, promoters therefore can be regulated by activators. How do the activators influence? By allowing the RNA pol to bind to the promoter region. So that way it will initiate transcription. Or by repressor, by preventing the binding of RNA pol to the promoter or weakening the binding of the RNA pol to the promoter. So let's say, for example, you have the DNA with the -10 and the -35, say, for example, and we know that within many of the promoters is present a cis element, that is a sequence of DNA, which is called as the operator sequences or the operator regions. And strategically, these operator regions are either placed within the promoter region or they are placed overlapping the promoter region, but they are associated basically with the promoter region. So, when you have the operator region within the promoter region, it has been observed that most repressors can come and bind to the operator region because they have a binding domain, DNA binding domain, that recognizes the operator sequence and with great affinity binds to the operator sequence. Now, when you have a protein already bound in such a way that it is overlapping with the promoter region, then the RNA pol will find it difficult to bind to the promoter region with great affinity. So, in a way, the presence of the repressor at the promoter region is going to act as a barrier for the binding of the RNA pol to the promoter with high affinity. And so, what is going to happen is transcription is not going to happen. Let's say, for example, consider another case where you have another cis element, but this cis element is recognized by the activator. So the activator comes and binds, and in presence of the activator without the repressor, you will find that the RNA polymerase is positioned on the promoter, and the positioning of the RNA pol enables transcription to begin. So therefore, in this case, you will not find transcription taking place, while in this case, you will definitely find transcription taking place. So this mode of or mechanism of action of the repressor and the activator is influencing the gene expression. Now, suppose you have a situation where you have both the repressor and the activator bound to the promoter region, then many studies have shown that generally it is the repressor that is able to prevent or act as a barrier to transcription. So even if then you have a repressor present and an activator present, the repressor has a greater say than the activator per se. So, but you must understand that if you have activators alone, then it increases the affinity with which the RNA pol will bind to the promoter region. So the question now is, how do these actually do that? You know, that repressors decrease the binding affinity and that the activators increase the binding affinity of the RNA polymerase to the promoter. So there is a concept that is called as cooperative binding, and this concept we have also seen in other molecular mechanisms. Say for example, during DNA replication, when one single strand binding protein comes and binds to a single stranded DNA, it will recruit many other single strand binding proteins to come and align themselves on the single strand DNA. Or, for example, you have DNA coming and binding to the 9 more regions, okay, which is the initiator regions in the origin of replication. The DNA once it binds to the initiator region, will enable binding of many more DNA to that origin site. So that is also cooperative binding. In other words, what is cooperative binding? A binding of one molecule to a DNA in this case, okay, allows binding of other molecules. So there is a cooperation between the DNA protein and also in cooperation between the protein protein. So you have basically together conformational changes that bring about what is called as effective or cooperative binding. So let us look at what we understand by cooperative binding. So say for example, you have the DNA with the promoter which has both the operator region and say for example, it has the activator sequence. That means the cis element for the activator bindings. And let's say for example, the activator comes and binds. So when the activator comes and binds to the cis element, there is a positive conformational change and that recruits the RNA pol to the promoter site. So you can see that binding of the activator to the DNA is bringing in a change both in the DNA as well as in the activator to actually make the RNA pol bind to the promoter with higher affinity. So that is what we understand. And therefore, this concept is what is called as cooperative binding, where the activator is ensuring that on its binding to the DNA, it is making the environment more conducible for the RNA pol to come and bind, or the conditions more conducible for the RNA pol to come and bind with to the promoter with higher affinity. Or you have another concept which is called as allostery. phenomenon which is called as allostery. This is also something that most would be familiar with, especially with respect to enzyme activity, where many enzymes have an active site, but other than the active site, it also has another site to which an inhibitor or again an activator come and bind. And when the inhibitor and the activator come and bind, it influences the active site, and thereby it would either slow down the catalysis or it will enhance the catalysis. So the same concept is basically over here. Allostery is the kind of concept that suggests switching on something and switching off something. So it's all or none. Okay. So that same concept can be seen even by regulatory proteins. That means they follow the same regulatory. The regulatory proteins can use this phenomenon as well for regulation of gene expression. So now let's say, for example, in case of allostery, it is the other way around. You already have an RNA pol that is bound to the promoter region and it is in a stable conformation, but it keeps the DNA closed. That it has bound to the promoter region, but it does not go from closed to an open complex. It just remains as closed complex. So it is a stable interaction, but it is again in a conformation that keeps the RNA pol in a closed conformation. Now suppose you have an activator coming and binding. The binding of the activator with the RNA polymerase initiates a change in the conformation of the RNA pol such that it goes from closed complex to open complex. So what is this? When you don't have the activator bound to the RNA pol, it is in one configuration, and when you have the activator bound to the RNA pol, then its conformation is different. So when it is not bound, it forms a closed complex. But when it is bound to the activator, it forms an open complex. So you can see a transition from one form to the other, and that transition is attributed to change in conformation of the RNA polymerase itself thanks to the allosteric interaction of the activator. If suppose you have, instead of the activator, a repressor coming and binding, then what can happen is that the repressor will change the conformation of the RNA pol in such a way that it will not allow the transition. It will keep the RNA pol as closed complex itself. So that is another way again of regulation. Here in this case, when the repressor is bound, it remains as a closed complex, but when the repressor is removed, it remains as a closed complex, but it can go to open complex. So with repressor, this is what is happening. The transition from closed complex to open complex is not allowed. Now, say for example, you have another condition, or rather what we have looked at so far is that most cases the activator and the RNA pol are adjacent to each other. So the binding sites of both the RNA polymerase and the activator are adjacent to each other, or binding of the repressor and the RNA polymerase are in proximity with each other. So far, this is what we have looked at. But in many cases, the regulation of genes are such that in those cases, the binding of the activator is very far off from the promoter region. So if this is the case, how is the activator going to, or the regulatory protein, how is it going to interact with RNA pol, say for example, to take it from a closed complex to an open complex? So what has been observed in many such cases where the regulatory site is very far off, you have the DNA looping. So this is what happens, and because it loops, the RNA pol is now interacting with the activator, and this interaction allows the RNA pol to escape the promoter and start transcription. The bending of the DNA can be because of certain proteins that are known as DNA bending proteins. Now, such a kind of thing is definitely observed in prokaryotes, but it is observed to a great extent for many genes in eukaryotes. So this basically is that action happens from a distance, and that happens because of DNA looping. So let us make the conclusions. In prokaryotes, most regulation of gene expression is at the transcription initiation stage, especially at the isomerization step of initiation. And there are certain mechanisms that also influence post-initiation, for example, anti-termination, or there are also mechanisms that uh, are carried out during elongation. But the most prevalent are the ones that happen at the start point of transcription. Activators and repressors themselves bind to the DNA and generally influence the RNA polymerase binding to the promoters, or also transition from the closed complex to the open complex. There are operator regions, that is cis elements present within the promoter region, or they are overlapping the promoter region, and therefore these become the binding sites for repressor proteins. And when repressors bind to these cis elements, then they act as barriers for the RNA pol to come and bind to the promoter region, and hence transcription does not happen or does not initiate. The regulatory proteins activate or inhibit transcription initiation by mainly two phenomena: cooperative binding and allostery. Thus, in prokaryotes are present regulatory proteins that can influence the gene expression depending on the requirements of the cell by using specific mechanisms. Thank you.